yaşıyoruz. İlk günkü heyecanımızla yılları aşıyoruz. Zaman akarken biz de kendimizi arıyoruz. Heyecanla, merakla. Sen de kendini mi arıyorsun? O zaman zamana yetiş. Bu sadece kendini bulmakla ilgili değil. Asıl heyecan kendini yaratmakta. Bir şeyler mi yaratmak istiyorsun? Mükemmel. Metali işlemeyi, bir bina yapmayı, bir iş kurmayı, hatta bir şehir kurmayı öğren. Hayat kurtarmak, teknoloji geliştirmek, üretmek. Hepsi burada. Ve burada tüm deneyimler profesyonellerle yan yana. Ancak hızlı ilerlemeliyiz. Çünkü bugün şimdi gerçekleşiyor ve yarın çok yakın. Hayatta öne geçmek istiyorsan önce zamanın önüne geçeceksin. Hep ileri gideceksin. Bu Doktor Suat İrfan Günsel. Tanıyor musun? Onun da söylediği gibi. Üniversiteler toplumların dokuma tezgahıdır. Toplumları geliştirir, dönüştürür ve gelişmesini sağlar. O burayı bugünden yarına hazırlamak için kurdu. Senin için, ileriye gitmek için. Yana ya da geriye değil. Sadece ileri. Çünkü gelecek çok yakın. Hadi harekete geç. Fikirlerin ne kadar ileride olursa yarına o kadar yaklaşırsın. Yanındakileri de geleceğe taşırsın. Bu senin geleceğin, senin fırsatın şimdi. Hadi yarını yakın. Hepimizi ileriye götür.
Welcome everybody. Today we will have a talk about the titled A New Era in Vaccine Development Leveraging Artificial Intelligence to Accelerate Vaccine Design. This presentation is organized by Operation Research Center in Healthcare, nearest University. And the talk will be given by Assistant Professor Dr. Zubaida Ameen and Assistant Professor Dr. Saleh Mubarak. Let me mention about them briefly. Dr. Zubaida Ameen received a bachelor degree in biochemistry from Bayero University, Kano in 2010, obtained master degree in bioengineering from Cyprus International University in 2016, and a PhD in biomedical engineering from nearest University in 2022. Currently a postdoctoral research associate at Operation Research Center in Healthcare, nearest University. She has established a robust track record in research as evidenced by extensive contributions to the field of artificial intelligence, biomedical engineering, and bioinformatics. Dr. Zubaida's research expertise is illuminated by her focus on the intersection of AI and healthcare, and applying cutting-edge computational methods to biomedical and environmental studies. Dr. Zubaida's skills are further exemplified by the application of various tools to complex problems in machine learning, deep learning, computer vision, data science, and data mining, with notable experience ranging from 3 to 4 years in each domain, which has demonstrated a remarkable ability to handle interdisciplinary projects, and has made several articles in prestigious international journals. Dr. Saleh Mubarak is an esteemed postdoctoral researcher at nearest University, specializing in artificial intelligence within the Operation Research Center in Healthcare. He holds a PhD in electrical and electronics engineering with specializing in artificial intelligence, a master's degree in electrical and electronics engineering, instrumentation and control from Sharda University, India, and bachelor's degree in the same field, electronics and control from Cape Breton University, Canada, and his research focuses on leveraging AI to enhance healthcare delivery and environmental management. Dr. Mubarak's pioneering work in AI applications has addressed critical challenges such as vaccine development and environmental sustainability. His contributions include the development of advanced AI-driven models for diagnosing and managing disease, significantly impacting healthcare and environmental studies. Recognized for his outstanding research, he received the Young Researcher Award in both 2021 and 2022, and Publication Award in 2023. And I would like to thank the organizing committee and our presenters for giving us the chance to learn about the vaccination design more. and stage is yours.
Right. Thank you so much, Ujam. Actually, yes, stand before you today to discuss the new era of vaccine development by leveraging artificial intelligence in order to speed up the development of vaccines. Today, with me, I have Dr. Zubaida, Ameen, which will be discussing more about the bioinformatics, and I will be focusing more on the artificial intelligence aspects. So, in this presentation, we are going to cover the background, introduction to artificial intelligence, the multi-epitope vaccines, the bioinformatic analysis, challenges, and conclusions. So, as we all know that in every society, vaccination is very important in order to reduce mortality rate, infection spread, and morbidity rates. So it is very important to develop vaccines. So in this developing these vaccines, we can say that it is costly, sometimes inefficient, and time consuming. And based on reports, it was observed that in developing vaccines, hundreds of millions of dollars to billions of dollars have been spent. So to cost all these things, artificial intelligence is very important in order to speed up the lab processes and time so that we can develop vaccines with very high speed. So we have different types of vaccines. For example, the inactivated vaccine virus, the attenuated virus vaccines, viral vector vaccines, DNAs, mRNAs, which is the messenger RNA, subunit vaccines, which are the multi-epitope based. We mostly focus on the multi-epitope based because it is more safer and easier to develop within the AI. So that is why we focus on that. So, what are the challenges of the traditional vaccines? Though the traditional vaccines shows or proven to be effective, but the time it takes to develop those vaccines is a little bit much, taking more than 10 to 15 years. Sometimes after you develop them, you realize that okay, the efficacy is not good. So you have to start over and over again, and you have to spend more on the lab, the culturing, and other stuff. So identifying suitable antigens that will trigger immune system is very important. So with the AI, it is easier to do all these observations and try to have the antigens that will be better and the protein sequence that are much more important, or the subunits of the protein that can trigger the immune response. So, as we all know that in developing vaccine, it requires preclinical trials, testing in animals, and testing in human population. So this also takes time, and it requires a lot of what am I going to say? uh, Time, and also there are a lot of regulations that we have to follow. So this is also affecting the process. So the reverse vaccinology started in the 1990s with genome-based methods, and also these methods use bioinformatics to analyze pathogens, genomes for effective vaccine candidate, and eliminating the need to use bacterial culture. So in the process of traditional vaccines, we have the pathogen culturing, the antigen extraction, in vitro and in vivo testing, and then candidate selection. So we want to reduce these steps. One, by using the genomic sequence, then we analyze the data and predict the epitopes that we want to use, which are important and can trigger the immune system. And then we validate the candidates. So in the steps in which AI enhances the epitope predictions, which are the most important aspects. You need to find the epitopes that are very important and then conduct the bioinformatic analysis. So with that, you can reduce the lab work, and also you need to also the validation of the vaccines, the epitopes using the in silico validation, the simulation, the web tools that we normally have, which are most of them AI based web tools. Okay, so this will speed up the process and it will give you more precise identification of the vaccine candidates, and it will lower the costs. So there are some vaccines that were successful and already been launched. For example, most of the major companies that are into drug have developed AI-based vaccines, be it the mRNA or the viral vector vaccines or the multi-epitope vaccines or that is the subunit vaccines. There are a range of vaccines, for example, for the HIV, malaria, breast cancer, and COVID-19. If you could remember during COVID, like vaccines were developed very fast. So these are these were developed by the help of AI. So let's all discuss about the artificial intelligence. I'll give a brief background on how it works before we can continue on the application in this particular area. So it started in the early 1950s and it started with the logic systems, then transformed into expert system. We have the era of neural networks, the machine learning, deep learning, reinforcement learning, and AI applications, which have been applied in many parts of our lives today. So we have several branches. For example, we have the machine learning, which under it we have the deep learning. If we look at the deep learning itself, it is embedded in natural language processing, speech recognition, robotics, visions, and all. So we can say that all these subsections they are interwoven together, and each of the systems based on the uh, are made based on the requirement of the problems that we want to solve. In expert system, it is more of like a planning as well, and finding a statistical reasoning and all. In speech recognition, you convert speech to text or text to speech. And also in vision, we have the image recognition, which is part of the classification problems that we normally do, and then the computer vision, which involve the detection systems. So in machine learning, mostly we have three layers: the input, the inference, and then the output. But mostly in machine learning, you are required to do the feature extraction or the feature engineering yourself. While in deep learning, you have layers and repeated layers of convolution, but you are not required to perform the feature extraction. For example, if you are going to use machine learning for image classification and all that thing, you have to extract the features yourself for each particular images, unlike in deep learning whereby the convolution layers will extract the information for you. So we have different types of problems. We have the regression and the classification problems. In regression, you go for a continuous value. For example, if you have a range of 1 to 100, you give a particular value, be it a decimal or an integer. While in classification, all you need to do is just to categorize based on a threshold to say that, okay, this is cold or this is hot. If it is below 50, maybe you can say it is cold. So this is just a brief introduction here. And as we all know, we have AI in all parts of our life, starting from the social media down to health, robotics, and finances, regardless of the discipline, be it sciences or social sciences. We always have AI. So one of the most important thing is data. Data is very crucial in AI because without data, you cannot the models cannot learn information to make decisions. So this data cleaning is very important. So there are different processes of handling data. We have the data cleaning, dimensionality reduction. You need to reduce the data so that it can fit well and can be trained in the model. And then you have feature engineering, whereby you work on the features that you want to extract from those information, and sampling of data, then transformation, transforming the data, the raw data into vectors, and then imbalance data. In case of classification problems, it is better to balance the data set so that you can have equal weights or unbiased weights. So in deep drug discovery, mostly we apply the AI at the starting stage, depending on the kind of data we have. We normally get the data from IEDB database and other studies that they normally conduct. So in this database, people do the lab works and then they deposited the epitopes there, but still you have the challenge of having different formats of data and variables that are involved in that particular data. So if you are getting the data from different studies, you have to work on it at first, um, like unify the data in such a way that you can be able to use it. And then after we selected the candidates, then we later on do the bioinformatics analysis. So in this study that we considered the tuberculosis, we got the data and then the tuberculosis protein, whereby after we train the model with the epitopes, then we apply a technique of sliding window on the whole protein sequence. We predicted the regions that has the that has the peptide sequence. After we did that, we filtered them out. Then we started performing the bioinformatic analysis in order to filter out the required ones. So, as we all know that the protein sequence is made of alphabets. So, normally we first pre-process it based on the on different encodings, because you can't be sure of the encoding that will work. You have to try all of them to see which one will give you a better result, which one will work well for you faster. Actually, for me, one hot encoding is one of the encoding that I really don't like, and it's one of the most efficient because it is time consuming. One, it requires a lot of resources, and maybe after 48 or 72 hours of training, you won't get what you want, and it is frustrating. So, but it's one of the best. We have level encoding and binary encoding, which we normally use them as well. But this is one of the popular and one of the most efficient, but highly costly. So, model selection. Model selection is very important because some models tend to work good with big data and some doesn't. And also depending on the model, if it is a linear model or non-linear model, and the characteristics of the data, you can see if it is a linear data or non-linear data. So it's more like a trial and error at this stage, but mostly we try to employ the machine learning models because they train faster. One, and if they did not work well, then we go for the deep learning models. And from experience, I realized that the recurrent neural networks, the GRU and LSTMs, they tend to perform better because mostly they are also being used in the speech recognition and all. So the model selection is very important. We need to select the models and then the model optimization. One, we have to optimize the parameters in these models. So you have to choose different parameters to see which one will converge faster, which one will give you less error, and which one will perform the task that you want to perform. So in deep learning, we have the normal optimizers like the Adam, we have the quasi-Newton optimizers, which are the LGBFs, And also we have the meta heuristics and bio-inspired optimizers. So you can optimize the models as much as you can, and then you try to see the parameters that will make the model converge faster. So the convergence is very important, and the less error you have, the better the model performs. This stage, it sounds easy, but really it is frustrating because you will have to try and try and try, and then after you try, then you try to get what you want. Because if you are working with a data of maybe 10 million sequence or 40 million sequence, so it is not easy for the models to train. And after you train, you have to optimize again to see if you can improve or not. And then later on, we have the validation. You need to validate the model. You have to provide the model with the data that the model has never seen before. And then you see the performance of the model to see how well it fits. So this is also very important. And mostly in this vaccine design, we just take the epitopes, we predict them, we try to find the optimal models, and then we select the candidates based on the sequence, and then later on we start the bioinformatics analysis. So I know it sounds very little, but really it saves a lot of time and cost. So mostly in this previous study that we are just focusing on, we try to use the machine learning models and the deep learning models, and also we use the recurrent neural network, the convolution, and the deconvolution, whereby we convolve the features and then deconvolve them back to the original aspect. The neural networks, which are the classical ones, random forests, Naive Bayes, and decision trees. So we try as much as possible to look at each and every option from machine learning to deep learning, and try as much as possible to get the best optimal models and the models that will give the higher performance. So this is what we normally do in the AI region. So I would like to give Dr. Zubaida the floor for the bioinformatics so that she will discuss more about that. Thank you so much.
Can you use microphone? Very.
Okay, before I continue, because we are going to be talking about the selection of epitopes, I would like to briefly introduce what these epitopes are. So in an antigen, there are specific regions on an antigen that we would like to use in order to develop these vaccines, and those regions are called the epitopes. Like we can see in the figure, like the blue part is an antibody, and the antigen is shown in red, and there are specific sequences on the antigen that binds to the antibody. So this region is known as the epitopic region, and it is what is being recognized by our immune system. So epitopes are usually like around 5 to 6 amino acid long, depending on the length of the region. And on the surface of an antigen, you can have numerous epitopic regions that you can select to use as suitable vaccine candidates. Based on function also, there are different kinds of epitopes. We have B cell epitopes and T cell epitopes. So what are the B cell types? They are the ones that bind to antibody, like shown in this figure. This is an antibody, and this is the antigen shown in pink. The red region that binds to the antibody is the epitope region, and we are interested in selecting the appropriate or the most suitable epitopes that will be used to construct the final vaccine using AI, because normally it takes time to do this experimentally, so we can speed up the process by using AI models. And this image shows the 3D interaction between this antibody and the antigen. Here is the epitope, and the interaction we can see as demonstrated in the 3D structure. And the other one is the T cell epitopes, which are also can be classified into different types depending on the type of major histocompatibility complex. And type 1 are usually present epitopes to cytotoxic T cells or CD8 T cells. And what they normally function is in killing the target antigen. And we have also type 2 MHC, which presents longer epitopic regions to T helper cells or CD4 T cells. So CD4 T cells or T helper cells will activate other immune cells that will trigger immune response. And the goal of vaccine development is to elicit this immune response so that our immune system can save them as a memory when next they encounter that same type of antigen. So in order to construct a multi-epitope vaccine, we want to incorporate all these types of epitopes, the B cell epitopes, the CTL epitopes, and the HTL epitopes into one single vaccine. And how they function is like the B cell epitopes are being recognized by the B cells, and that will activate the plasma cells to produce antibodies. Similarly, the CTL epitopes and the HTL epitopes will also become activated, and they will produce chemicals that will attack and kill the infected cells or cancer cells in the case of cancer vaccines. So together, this cellular response and the humoral response will lead to a strong immune reaction that will trigger immune response that we want in the target vaccine. So the advantages of multi-epitope vaccine, as we can see, one is they composed of different types of epitopes, the CTL, the HTL, and the B cell epitopes. And based on that, they are more efficient than using just a single type of epitope-based vaccine, because there are some B cell-based vaccines or T cell-based vaccines. But when you combine all these type of epitopes, you will have a stronger immune response. And also you can, since you are selecting the desired epitopic region, you can remove unwanted component or toxic components of a virus. So that makes multi-epitope vaccines more safer than traditional based vaccines. And also you can combine multiple epitopes from different virus. For example, you can design a multi-epitope vaccine against tuberculosis, and at the same time, it will work on SARS-CoV-2 if you mix the different epitopic regions from different virus. And the same also for tumors. You can develop a vaccine that can target various cancer cells, not just one particular cancer cell. So we can see they are highly efficient. And this is the next generation types of vaccines. You can also add other components, like you can tailor them to your own design. You can design them the way you want. You can add different types of adjuvant to improve the immunogenicity of the vaccine. You can add different types of linkers. You can add different stabilizing compounds to your vaccine. So you can design them to your own function. So these are the advantages of multi-epitope-based vaccines. So in this case study, I'll be talking about the multi-epitope vaccine that was developed for tuberculosis. And as the first speaker already explained, after we've trained the machine learning models, we use some specific proteins of the Mycobacterium tuberculosis. And these are the accession numbers of the proteins. We use these proteins and we send them as an input to the trained model and ask our model to give us the sequence that will be suitable as an epitope in our vaccine. So first we asked the model to predict the B cell epitopes, and it gave us some sequences. But we did some validation tests or analysis to check if these epitopes predicted by our models were surely going to be B cell epitopes. So we can do that by using other bioinformatic tools like BepiPred, BCEPred. So there are various available tools online. then you can check the B cell epitopes of your sequences. So it was observed that our vaccine really contains B cell epitopes. And we also did the same thing for the CTL epitopes, that is the cytotoxic T cells. And we also observed that our epitopes contains different types of cytotoxic epitopes, like this sequence has 8 cytotoxic epitopes, and these are their subtypes because MHC molecules are very diverse. You have different alleles or subtypes. So it shows that the epitopes that were predicted by our model is going to have a strong immunogenicity based on the composition of the B cell and CTL epitopes. And we did the same thing also for the HTL epitopes. We used a web tool to confirm whether the sequences predicted by our models contains HTL epitopes. And as we can see, they also contains HTL epitopes, and these are their various subtypes. So finally, before constructing the whole vaccine, we have to do additional screening. Like we have to check for toxicity, allergenicity, and antigenicity. So we don't want to use a sequence that is going to be toxic, and also, but we don't want any allergic reaction, but we want the sequence to be highly antigenic so that it will generate a strong immune response. So in this table, we can see that all these sequences are non-toxic. We use a web tool known as ToxinPred to predict the toxicity of the epitopes. And also we checked for their allergenicity, and none of the sequence shows any allergic reactions. None of the sequence we use in the vaccine has any allergic reactions, but at the same time, they are antigenic, so they are going to generate a strong immune response, and that is the goal of our vaccine. So we use also another tool, Vaccigene, and we can see based on the scores, they have strong antigenic property. So finally, this is the sequence of the whole multi-epitope vaccines. We then join all these epitopes that we predicted with our models using appropriate linkers to connect them together. And as I've mentioned, you can add some components to your vaccines. And at the beginning, we add an adjuvant, which will increase the immunogenicity of the vaccine. We use the glyceramycin, which has shown to be effective against tuberculosis. And we use other different components to different linkers to join the epitope sequences of our vaccine. And we added a polyhistidine tag at the end of the vaccine for easy purification or identification. So in a summary, this is the structure of the constructed vaccines. The boxes shown in gray are the B cell epitopes, and those in pink are the HTL epitopes, and the CTL are in yellow. And we link them using the different types of peptide linkers. But at the beginning, we added an adjuvant. So this is the architecture of the vaccine we designed. Then we also further check for the 3D or tertiary structure of the vaccine, and it shows it has a good structure. It has random coils, it has beta sheets, and it has alpha helices. So it is going to be highly antigenic. It's going to generate strong immune response. But we wanted to see really, is it going to be recognized by our immune cells? So we also conducted molecular docking using some bioinformatic tools. And we observed that there is going to be binding, strong interactions between our multi-epitope vaccines and the receptors. So this shows that we use TLR receptors. So if our vaccine is going to bind to the TLR receptors, it means it is going to bind with our immune cells. And we also checked whether the vaccine is going to be expressed in living cells. So we cloned them using a plasmid vector in E. coli cells, and there was high expression in E. coli cells. So meaning it can be expressed in living cells. Finally, we check what is going to happen if we inject the vaccine into human body, and we perform this immune response simulation, which is very important. First, we decided to use 3 injections, as we can see in figure A. The black line is the antigen, meaning the vaccine, and we can see after first injection, the amount of the antigen increases, meaning our body has responded and decreased and attacked the antigen. But even though there were small differences between the second and third injection, the injections were stays within 2 months, but we observed production of strong immunoglobulins. And as we have said that our vaccine contains B cells and T cells, you can see in B cell populations in B, the B cell population increases after the primary and subsequent immunizations, and also the T cells increases after first and second dose of the vaccine. And the same thing with the cytotoxic T cells, there was increase in their population until it reaches around 150 days. And we've also seen increase in macrophages, which are important for antigen presentations. They are going to present the vaccine to the immune cells. So there were high macrophages and also high production of cytokines, which will activate the immune cells to come and attack the foreign antigen or the virus. So, and this shows that our vaccine is going to produce strong immunological response so that when it encounters the virus, subsequently, it is going to attack and kill the virus cells, kill the virus and also eliminate the infections. So finally, the challenges of this work, like the first speaker explained, is the data quality, like the type of data you have, and secondly, the computational resources, what kind of computers or machine are we using to do the work, because when you're dealing with sequential data, it's going to take a long time. And also different lab results sometimes deposits different results in different formats, so it's not that easy to normalize and arrange the results in one format. So these are some of the challenges of this research. But in the future, we can see that with this type of vaccines, we can design personalized vaccines. You can tailor a vaccine according to the function you want it to carry out. And also that will help in controlling outbreaks, especially during pandemics. And these are some of our published articles. We worked on the vaccine against tuberculosis and COVID-19. And also we use an explainable AI also to design the COVID-19 vaccines. Thank you for listening.
Dr. Saleh Mubarak and Dr. Zubaida Ameen, thank you very much for this nice talk. And we have great information about the design of the vaccine and how it accelerates the producing vaccine compared to a classical model, I guess. So is there any question?
Thank you for this very nice presentation. It was very informative and impressive how the AI technology is developing right now. So I have a couple of questions. In terms of selecting the epitopes, you said you selected you picked the epitopes from the open source database from the internet. So how did you filter out the specific epitopes against specific diseases or cancer or any infection? Likewise, we did in the tuberculosis application. This is one of my questions. And my other question is, it is very impressive that you can identify whether a single dose would be enough or like triple dose should be required, or you can monitor the CD4 positive T cells behavior upon macrophages, etc. And you can even identify the polarization, like whether it's going to be Th1 or Th2 response. So I'm just wondering if there is any other study which is correlating, like which use this AI based design to design a vaccine and actually they produce the vaccine and did in vitro, in vivo experiments, and whether these two studies, like in silico, in vitro studies are correlating with each other. Like if there's any study like this, if you know, and give example, I would be very happy.
Okay, thank you for your comments and questions. You've asked a couple of questions. First, I want to start with of course, there are some vaccines like the peptide, the multi-epitope vaccines that are still under clinical trials, but also we have some vaccines that are peptide-based and have been approved.
These are produced with the help of the AI-based approach, or?
Yes, there are those that were produced using AI. Most of them are still under clinical trials, but most of the ones that are produced here, they are mostly different types of vaccines that, but they are peptide based, but they are still available. Like we have here, these are mRNA vaccines. We have a couple of examples that are peptide based. In this case, the breast cancer vaccines and this one are peptide based, right? So there are also some, though they were not included in these presentations, but they were produced using the artificial intelligence tools, and they are under clinical trials. And there is a vaccine against COVID-19 that has been approved for use in Russia, and it's a multi-epitope based vaccine. So I cannot remember exactly the name. Another question? I cannot remember. Yes, you also asked about the immune response. So this it was a simulation. It is not like experiment in the lab. It's a simulation, but it is going to give us an idea how the vaccine is going to interact in the human system. And also you can adjust the settings from the database. That we use default settings, but if you want, you can change. Like you can see, you can use 1 injection, 2 or 3, depending on what you want to check. We've worked with 1, 2, and we decided to go for 3 and see how the vaccine is going to interact with the human system. So, but it shows really that the vaccine is going to generate strong immune response. Even with 1 injection, it was successful. But we showed for the 3 injections. And also you've asked about the data, how we collected the data from the database. If you go to the page, the database, you can select what you want. You can select the type of microorganism that you want.
Is it specifically disease or?
It's an immune epitope database, IEDB. I mean, it's specifically for epitopes. You can have different types of epitopes depending on the type of microorganism.
You have an option to select the
Yes.
specific microorganism.
You have an option to select specific microorganism and also to select the type of epitopes you want, whether B epitopes or T cell epitopes. You can select from the page.
Yeah. What was the reason of working on tuberculosis?
Because tuberculosis, it's one of the infectious diseases that causes death worldwide. And also it is a very strong disease, especially during COVID-19. People with tuberculosis and COVID-19 tend to have less survival rates. So we decided to see if we can generate an advanced vaccine, but a more like advanced vaccine, because a peptide-based vaccine is safer than traditional based vaccine, especially for immunosuppressed patients, those that have weak immune system. So we can tailor this type of vaccine to them because it's going to be safer. It has less allergic reaction, less toxicity and all.
Can I ask why there is not any idea? Maybe there is the idea of the generating the vaccine for cancer.
Yes, we are currently working on some cancer-based vaccines. Currently, yes, we can develop. Yeah, it's possible, and we are working on that too.
Okay. Thank you very much for this wonderful presentation. Actually, I have two questions, but one of my questions is what it asks about the source of the proteins and how you go about answering. You made mention of docking where you use E. coli, which is a gram-negative organism. So my question is, why did you decide to use gram-negative organism in testing the antigenicity instead of incorporating both gram-negative and gram-positive? Why did you only use E. coli? Go to that slide. You made mention of using E. coli.
Yes, this one.
Yes.
This was for checking for expression in a living organism, but for the interaction, like the docking, it was using this some receptors, the toll-like receptors, which are peptide-based receptors. But for expression in living cells, you can choose E. coli, which is the most suitable organism that is mostly available for this simulation. It depends on the tool you are using. So in our tool, we normally consider, in this tool, it is E. coli that is normally considered. So, but you can maybe check with another microorganism.
Yeah, as
It's
warriors and other aspects, they are more, I can say, more rampant with this one. Okay, that's by the way. Then the next question is, this is on paper. Now, if we, after producing this vaccine, and we decide to use it, do you think there will be consequences? And if any, how do we go about to contain it?
yeah,
Because
that
this is just on paper. So can we go to the lab and do something about this?
yes. Before this is like you are doing in silico experimentations. Then next you have to do preclinical testing. Whereby you have to test it in experimental animals. Then the experimentation is in stages. Before you proceed to clinical trials, and also that has like stages. You have like 3 phases: phase 1, 2, and 3. So any vaccine that has to be manufactured doesn't directly goes from production to the human body. It has to pass through these stages of screening. So I believe before then, if there is any issue, it might have been resolved by then.
Producing separate type is costly or not?
Normally, this type of vaccine, peptide base, is not costly because you are synthesizing the peptides chemically, so they are not very costly. Yeah.
Well, thank you so much for your wonderful presentation.
Thank you.
We learned a lot, more especially when you're presenting the chemistry parts. Actually, like, you give a lot of details about it. My question is that you recently said that you're working on cancer-based vaccine. From what I observed, actually most of the viruses actually usually present antigen. And it's from that antigen you can really work on the epitopes. So with regards to cancer, majority of cancer cases actually if they are genetic. So how will you go about predicting the epitopes from cancer disease?
Yes, first, thank you for the question. So in the case of cancer, we are definitely going to look at the epitope regions, because even on the cancer cells you have proteins, like, so our target, since it's a peptide-based vaccine, it's a protein. So we are going to target the proteins of the cancer and then select the regions that we will use to construct the vaccines from the cancer cells. We are going to remove, like, from the proteins of the cancer, the regions that we want to incorporate into the vaccines.
So we have a hope for the future.
Thank you very much for your attendance and for your time. Thank you.
Güzel resim alabilir miyiz komputer? Evet.
Hayat hızla akıyor ve biz bu hızı seviyoruz. Her güne heyecanla başlıyor, her anı doya doya yaşıyoruz. İlk günkü heyecanımızla yılları aşıyoruz. Zaman akarken biz de kendimizi arıyoruz. Heyecanla, merakla. Sen de kendini mi arıyorsun? O zaman zamana yetiş. Bu sadece kendini bulmakla ilgili değil. Asıl heyecan kendini yaratmakta. Bir şeyler mi yaratmak istiyorsun? Mükemmel. Metali işlemeyi, bir bina yapmayı, bir iş kurmayı, hatta bir şehir kurmayı öğren. Hayat kurtarmak, teknoloji geliştirmek, üretmek. Hepsi burada. Ve burada tüm deneyimler profesyonellerle yan yana. Ancak hızlı ilerlemeliyiz. Çünkü bugün şimdi gerçekleşiyor ve yarın çok yakın. Hayatta öne geçmek istiyorsan önce zamanın önüne geçeceksin. Hep ileri gideceksin. Bu Doktor Suat İrfan Günsel. Tanıyor musun? Onun da söylediği gibi. Üniversiteler toplumların dokuma tezgahıdır. Toplumları geliştirir, dönüştürür ve gelişmesini sağlar. O burayı bugünden yarına hazırlamak için kurdu. Senin için, ileriye gitmek için. Yana ya da geriye değil. Sadece ileri. Çünkü gelecek çok yakın. Hadi harekete geç. Fikirlerin ne kadar ileride olursa yarına o kadar yaklaşırsın. Yanındakileri de geleceğe taşırsın. Bu senin geleceğin, senin fırsatın şimdi. Hadi yarın yakın. Hepimizi ileriye götür.